So very much thankful to the Prakara for giving this platform to sharing my knowledge to the students, and students are benefited. We are one of the pillar for the giving the enlightenment to the students. So we are happy to take this session actually. Thank to Prakara giving the opportunity to me to discuss with this session with the students. I think I introduce myself. Myself, Vinod Kumar. I'm assistant professor in MMM College of Health Science, Chennai. I'm taking it subjects hematology, blood bank to my student. I'm teaching actually. In past 10 years. So I'm going to talk about the topics today is about blood grouping genetics and pre-transfusional investigations. First things about that, what is blood group? You already know about. Blood group is important to us. Before we go for a blood donation, doctors asking you that what is your blood group. It is a common thing now. You can see in the your identity card itself. One of the mandatory thing is your blood groups. So now, as a technologist, we have to understand about that. What is the importance of these blood group? Why they're giving a priority to place into the ID card as a blood group system? So now we're going to talk about the what is the blood group, how the blood group is going to be inherited from parent to the children, as well as a technologist, you should know about that. Blood group system is very important in the blood bank. But as a blood bank technologist, how you people do some investigation in the towards to the blood group systems. Like before giving a blood transfusion, okay, you must to go for a what pre-investigation. The pre-investigation called before giving a blood, we have to do the investigation. That is called pre-transfusional investigation. We're going to discuss about the topic now. So this is a synopsis I'm going to talk about in the 45 minutes about the blood group and blood group genetics, which include the inheritancy, formations of grouping, what is ABU antigens, what is Bombay Bell group, what is secretors, non-secretors, what is RS typing, and the second thing is about that investigation oriented. As a technologist, we are have a very important section about in that blood groups systems and RS typing. What are the methods we have in the laboratory? That is we're going to discuss. And along with that, the main important thing is before transferring the blood to the patient, we matching the blood with the patients with the donor, and this is going to be called as a cross matching. So cross matching is going to be considered about the what compatibility of each other. Another thing is about that we're going to talk with another thing, Coombs test. So Coombs test is generally called as anti-immunoglobulin test, which is widely used for the confirmatory of the compatibility. Come to know about that any specific antibodies present in a patients or donor, we are finding through by this method and ruled out the problem of transfusional reactions. So it's a mandated test for at the hospitals. The last thing is about that antibody panel screening that will give the enough knowledge about that. What are the antibodies we supposed to have in the blood systems? How it is going to be affect the patients? So these are all going to be considering about that antibody screening investigations. Okay, let's we move on to the first thing, blood grouping genetics. Okay, so discovery of blood group systems. So the first blood group system was discovered by the Karl Landsteiner in 1901. He awarded a national award 1930, and he investigated about the ABO four group systems in the blood, and is one of the best inventor in the entire people. Now we are talking about blood groups and everything for transfusion. Now it is helping for the people to make alive. Okay, he is one of the best person. Okay, next thing is about that the inheritance of blood groups. So the blood group is how it is going to be forwarded to the children from the parent. That is very very important. It is given by the Perstein in 1924. He gave the idea about that we have a chromosome 9, the chromosome 9 which carry the or ABO grouping. Okay, the main thing is about that ABO grouping is carried by the important thing is called ABO gene. Okay, we can see that actually our parents are giving the two hand. One is from your father, one is the mother. Same way, the father and mother is helping you for your blood groups. One gene is given by your parent, for the father, and one gene is given by mother. And this is going to be called as a what? Inheritance of your blood group is carried by the children, by the parents. Okay. Let's we move on to the next thing about that genetics. As you know about the genetics, the basics, I'm just give some outline. The chromosomes. Chromosome is the main thing about that. The information center is going to be called a genomic information center for the DNA. Okay. What is gene? Gene is nothing but is going to be called what? Any information we have, that information inheritance which is going to be positioned in the chromosome. That is going to be gene. We always tell about that any characteristics present in the chromosome, we are going to locate some area. That is going to be called as like address. If you want to ask address about anybody, you're telling the address. Same way, if any blood group we have, the blood group antigens where it's supposed to be located. The location area is called what? Locus region. We always tell about the one point about that triad. Triad is nothing but we individually we have one characteristics. What characteristics I have, that's my triad. Okay. The next thing is about that allele. So what is mean of allele? So allele is nothing but simply called as here. A parental gene will help you for your making your one characteristics. That is called parental character, father character, mother character, and both the allele is going to be help you for to enhance your own characteristics. This is going to be allele. And here we have example of allele is dominant allele. The dominant allele is father, mother, both is having a dominant gene. Example I'm giving you here actually A gene, O gene, which always give the group of O to the children. Co-dominant. The co-dominant is both the parents are having a very dominant gene like A and B. Always they expressing the dominant same both characteristics A B groups. If go to the third thing is about that the allele is going to be a recessive, that is called what suppressive characteristics O. If it is going to be a suppressive characteristics, and finally the children will get all these O groups. So this is about the blood group genetics we generally seeing about that, and how we are connecting these genetics into the our blood group systems. Let's see about now. We have the I told already parent is giving you the allele. Okay. One is called heterozygotic allele. Next thing is homozygotic allele. So what is mean of heterozygotic allele? If parent is group A gene is giving you, if one parent that mother is giving a B gene to you, the A gene B gene connect together going to be called as a what? Dominant allele. Okay, and this dominant allele is going to be a different type. One is A, one is a B. Is called as here heterozygotic allele. Okay, if you say that actually, what is mean of homozygote allele? Homozygote means have the same characteristics. Example, if the parent is A, my father is A, if the mother is A, you can see that the baby born with the A group. And this is going to be called what? Same gene. And this is simply called as a homozygote allele. So, the maximum blood groups which is going to be given by the children by the help of the parents. Okay. Here we're talking about the one important thing is about the genotypic character, phenotypic character. Always you come to know that though the parents are giving their own gene, but you have a own point of acceptance. That means, if I told already, example, if the father is A group, if the mother is a O group, and you are getting an O group, and this is going to be called what? You are complementary. What complementary is? I'm supposed to produce the own characteristics. The simple call as a phenotypic characteristics. So, what is mean of genotypic characteristics? If the parent, if father is giving a gene, if the mother giving a O gene, I am producing my own characters called genotype. What parental groups I have, that is called as a phenotype. Okay, I think you got a clear idea about basic genetics. Now let's talk about the ABO's group system. Antigen is segregated into the two different region of your blood systems. One is antigens in the RBC, antibodies in the plasma. Let's see about that. What are the blood groups we have? One I told already: A groups, B groups, O groups, and AB groups. Let's we talk about the first group about the A. You can see that A group is having a genotype. I told already A always have a genotype and phenotype. So what is mean of genotype? I told already. A one from parent, mother, and father. Let's say first example. If father is having a A gene, mother is having a A gene, and the children is going to be have a one character that they call A character. So the baby has to produce the A groups. Okay, and finally we can see that actually the antigens in the outside of RBC, A is there. Another one more supporting antigens called what? H antigen. So the H antigen is a very important precursor antigen to preparing for a blood group systems. That is called base. Without the base of H antigen, you cannot able to prepare any kind of groups. So H is a very base for everybody. So you can see that the A group is also having one more additional antigens called what? H antigen, that is simply called as a AH antigen. And these people having this antibody called what? Anti-B. So always see that A group people having a antigen, H antigen, and B antibody. In the case of AO, if the parent is having this is going to be called what? Heterozygotic genetic character. That we call A and O, different. Okay, we'll see that actually the baby giving us a report of A group, he'll say that same A antigen, H antigen, and going to be a antibody of B. Okay, this is going to be a segregation of antigenic character for the A groups. Looking to the B group, same. The B group is going to be a BB if the genotypic is going to be a homologous. If the baby having the group of B, the baby is supposed to have the antigen for B as well as H. Also, we can see that the plasma of the baby having the anti-A. Let's go and talk about the O groups. What is the O group? Let's see the picture in the O group. There is no antigens present, but it's have the antigens called what? Only H antigen. So O group, you don't say that they're not having any antigen. They are having antigen. They are having antigens called what? H antigen. But they are not having the O H antigen. After that, they are not having a antigen, B antigen. That's why we are calling as antigenic carriers. Okay. Let's now talk about the H antigens who have in the blood system. Definitely, they are called as a O groups. You can see that the O group is having a genotype is O, going to be a that we call homologous recessive gene, which is carried by the parent, and the baby giving a O groups. Finally, the group is going to be called O. So they're having the antigen H, antibody A, antibody B, both is present in the children blood plasma. Let's go and talk about the last one, is called AB groups. If the person is AB groups, you can see that genotype is AB, going to be heterozygotic allelic characteristics, and the baby always form a two groups as A and B also. So that's why called AB groups. So here you can see that actually the baby RBC outside, you can see that there is a three antigen which is present. It's called A antigen, B antigen, and H antigen. And you can see that there is no antibody which is present in the AB groups. Okay, so this is going to be called as antigenic characteristic, which is going to be distributed to the each different groups. Okay, we're going to talk about now about the inheritance of blood group, how it is traveled to the parent to the children. Let's see the first example. I'll show you that if mother O, father O, possibly the blood groups of children is going to be O. And what are the impossible group of the children is A group, B group, and AB. Let's go to the second example, O parent and A parent. The children is going to be, have a chance of O groups and A groups, and it's not a possible for a B groups and AB groups. So this is going to be called as a what? Inheritance law. The inheritance law, which is given by in detail by the what? The important person Mendelian law. The Mendelian law, it is telling you about that how two different kind of genes fighting together and expressing their own characteristics. So this is why I'm telling you that actually the parent groups, whatever it may be, how the children is expressing the own character, this is going to be simply called as a inheritance. They are presenting their own characteristic called what? Possible thing is going to be a phenotype. If father, mother, what they have, that is called what? Genotypes. Okay. So this will help you to that. What are the possible groups? What are the impossible groups? We learned here actually. We'll see in the case of in the last one actually. If parent is AB, if father is having, the mother is AB having, there definitely we can see that actually all the possibility of children have A group, B group, AB group, except the baby does not have the group of O. Okay, this is going to call as inheritance, which is not allow for the O group into the possibility of AB parents. Okay, let's talk about the important thing is I told already why we need these blood groups, why we should have to add into the blood our ID card as a blood group is important. So here we see that you are different groups is here actually. So we can see that if person is a O positive, O positive people are getting the blood from the positive people. That's called they can get the blood from O people, A people, B people, AB people. So we can see that actually O is donor for everybody. Also, O is only restricted for the giving a blood to some restricted people. Okay. Next, to go for a negative people, see that actually O negative people have a lot of option for everybody. So the problem is actually O negatives have a more chance to give everybody. They call simply called universal donor. So you can say that universal donor is O negative, but you can see that the ratio of O negative is very less. So that's why they always depending the what O positive people to give the blood donation to the patient. That's why we are calling as a O positive as a universal blood donor. Let's come to about that the point of AB groups. We see that AB groups have a possibility of all acceptancy. They can receive blood from everybody. So this is the phenomena called as a what people are accepting blood from anybody called what AB or acceptor. So we can say that recipient who are recipient AB or recipient they can accept the blood from A group, B group, AB group, whatever may be. The one important phenomena is positive can give to the positive, negative can give to the negative. This is one of the important concept you should understand here. Let's we have the topic about that formation of ABO grouping. How the ABO grouping is forming. It's how the parent is giving a gene. How we modified a gene into the A group, B group. One important thing is we call what we are going to have a genetic characteristics H. So and this is going to be heterozygote H and homozygotic H. Okay, so this is going to be give you the what the important thing is formation of H antigen. So formation of H antigen once if you are forming, you are able to produce the what chance of getting the A gene and B gene. So A gene B gene is always dependent to the parents. Whatever the parent is having a gene, it is giving you that gene, and you are try to prepare here some kind of specific enzymes. So the specific enzymes which is contributed by the special genes that we call A gene, B gene. Okay, now we're going to see about how the chromosome 9 will help you for the presenting A group, B group with help of H antigens. Okay, I think this is okay for you actually. All of you, this is a hello. Sorry to interrupt you. Can I continue? Hello. This is the formation of ABO group antigens. Okay, so let's see the picture actually. The picture is telling about how antigens are sharing into that every RBCs outside, and now they're supposed to be blood group systems. Let's see the first thing is about that. How people are forming the H antigens. Say H antigen is going to be produced by the H precursor molecules. Example RBC on the top, we're going to place some chemical. So this is going to be a glucose, galactose, N-acetyl glucosamine, and galactose. Finally, we'll have a lactose. If you make this all complex, this is going to be called what? Chemical preparation. If you are preparing this chemical into the H surface, what will happen? It has become an H antigen. Let's see about that. Once if you are forming a H antigens with the help of all these chemical molecules, you become in the absence of for what? Getting a antigen. How the people are getting a antigen? If the person who's having a gene from the parent, if the parent is contributing any agent to the children have the ability to produce the one extra chemical. The chemical called as a what? N-acetyl galactosamine. And this N-acetyl galactosamine, which is produced by the A gene, we can see that there is a formation of A groups. So how the A group is forming? With the help of H antigen, the parent is giving a one important amino acid complex called what? That we call enzyme transferase N-acetyl galactosamine, which is going to be placed into the H gene. It is modified as a A gene. Okay, the simple concept is adding extra molecule into the H antigen. It has become a form as an A group. How the B group is forming here? See the third picture in the bottom left side. The formation of B antigen is supposed to see that actually I am just taking up the N-acetyl galactosamine, replacing the galactose. If I replace the galactose, what will happen? Automatically it is turned to the next group, called what? B groups. So this is going to be called what? So there is a difference between A group, B group. Only thing is called what? One complex is interchanged. One is called N-acetyl galactosamine is present. It is going to be an A antigen. If the person is who's having the replaced with the galactose, that is going to be called as a B antigen. Let's we talk about the fourth picture. The fourth picture is about that Bombay O group. So what is mean of Bombay O group? See the clarity between the H antigen in the first one picture. See the law for fourth picture. The first picture itself, we can see that presence of galactose. You can see that any person is missing the galactose. Okay, the people called as a what? H suppressor. People not able to produce the H antigen. You can see that Bombay O group people not able to produce the H antigen is simply called as a H lag people. They are not having a ability to produce the H antigen. Okay, this is going to be H suppressive characteristic people. Okay, let's now see about Bombay groups. After that, we're going to see about the antigen systems of blood groups also having the subgroup systems. So we see here actually the subgroup systems of ABO group. We have one important thing is A1, A2, A1B, A2B. And you talk about any person is telling me about that I'm a A group. If you are A group means what are the your subgroup characteristics? If the subgroup characteristics is going to be a A1 or A2 or A1B or A2B. We see here actually 80 percentage of people belong to the A group or considered as a A1 category. 20 percentage of people consider as a A2 category. Either it is a A1B or A2B, whatever it we take. A1 is probably 80 percentage of people occupied groups. And remaining groups are going to be A2 groups. A2 groups are only the 20%. We'll see here the difference between the each categories, antigen, antibody, which is going to be distributed. Let's talk about the first characteristics, A1. If the person is a group of A, what are the antigens he is carrying actually? He is carrying the A antigen, A1 antigen. The antibody is supposed to have in the plasma anti-B. So this is going to be a group of A1. We'll see about here actually the A2 groups. The A2 group, we'll see the second one. A2 group is having the antigen of A is supposed to have the additional extra antibody called what? Anti-A1. Always think that actually A2 is not having a chance of getting A1 antibody. But here you can see that A2 groups should be have the extra additional one more antibody called what? Anti-A1. And also we see here actually A2B is not having a chance of anti-A1. We'll see that A2B is going to be have anti-A1. So anti-A1 is supposed to be present people who are having the absence of A1 antigens. So A1 antigen absent people, they are replacing the antigen antibody in the plasma. So here what are the problem is actually, see that actually if A1 is a patient, A2 is in a donor, if the A2 donor is giving a blood to the A1 the patient, definitely you have a chance of getting anti-A1 is going to be reacting with the A1 antigen and the A1 is getting a chance of reaction. But this thing is won't be reacting because of this insignificant antibody. What you will see that actually people are A2 giving a blood to the A1, there is a maximum insufficiency. That is called there is no chance of getting the hemolysis. There is no possibility of getting blood transfusion reaction. That's why they are allowing A2 group to give a blood to A1. Okay, these are the conditions. Okay, let's see about the next thing about the Bombay O groups. So the Bombay O group simply called as the OH phenomena. Here you can see that actually I told already H antigen is who's try to produce always called as a O people who are not having this H antigen producing characteristics simply called as a H recessor. simply OH phenotypic characteristics. So this is invented by the Blandy in 1952. He informed about that people having the O but not the H antigen. O missing of H antigen simply called as a Bombay O group people. See the picture here actually. So we'll see that H is an important substance to create the ABH system. I told you the ABH group, A group, B group, whatever you take, H is one of the important complementary characteristics. But when you come into the category of O, Bombay O group, you see that there is an impossibility of producing the H antigen. Missing of H antigen, the people are called as a what? O phenotype characters of Bombay O groups. And these Bombay groups having the one problem is about that they are having the H antibody in the plasma. That is very, very important. None of people having the H antibody, but only the people of Bombay O group having the H antibody. This is very, very important. O normal group, Bombay O group. If the Bombay O group give the blood to the normal group, they are getting the problem of reactions. So here the problem is about that only Bombay O group can give the blood to the Bombay O group. So this is going to be important condition about that. Bombay O group is not getting blood from other people. Only have a chance of getting their own groups that we call Bombay O groups. Now let's see about another thing is about that. Always see that can I do my blood group systems apart from the other body fluids? Yes, there is a possibility. That's why we are having about the secretory genes. So we have a secretory genes here about chromosome 19, which carry the gene SCE gene from the parent. So if it is a dominant gene we are having, definitely we are trying to have this antigens of ABO, which is going to be diluted in the plasma, gastric juice, saliva, sweat, tears, semen, and milk. So we can see that actually 80% of people genotypic character SE, which is going to be have a presence of all these ABO antigen diluted in the plasma. We always see here actually antigens always present on the top of RBC, but the thing is actually in the case of secretory genes who are having the antigens supposed to also diluted in the liquids. This is called liquid antigens. So this is going to be expressed by the genetical characteristic. We'll see the first the box actually. Genotypic inheritance. If the person is a AB, you can see that they have the HH gene. You can see the secretory gene is going to be a too dominant. Always see that the person is able to produce the ABH antigen. Also, the ABH antigen present in the saliva. So this is going to call it as a what SE gene genotype is which promoting the what presence of antigen also in the liquid fluids. You're talking about the second one is the AB group and HA. You can see that is going to be a recessive characteristic of secretory gene, and these people only having the ABH antigen in the RBC, absence of antigen in the saliva. So in the liquid body part of fluids, which not having the antigen, this is going to be called what non-secretory genes. So we have a option for us actually one secretory gene, non-secretory gene, people having ABO group in the RBC, also ABO group in the body fluids, and maximum people 80% of people having the blood group antigen also distributed in the various body fluids. Let's we talk about the RH systems. The RH system is important for us actually come to know about we are a positive people or negative people. Hear about the Stetson and Levin and Wiener, Landsteiner, both poor people. They are giving the largest idea about that. What is the RH blood group systems? And see that actually 90% of people we are all positive for the RH positive. Okay, and only the 5% of people belong to the negative. Let's now see about that. What is RH positive? What is RH negative? Yes, if person who is having the any additional antigen rather than that ABOH, I told you ABH is a general blood group system. Here is about that D antigen. So if any person is having an additional extra one special antigen, D on the surface of RBC is going to be D antigenic carrier people. So these antigenic D carrier people simply called as a positive people. Sometimes what you will see that actually these D antigens going to be very weak in the RBC top. This is going to be called as a weak D people. So weak D people means people who are having a D antigen. The D antigen is having a very lower quantity on the surface of RBC. So these people you can try to find by the important test called what ICT. That's simply called indirect Coombs test. This is simply called as a DU testing. If you do the DU testing, we try to confirm the people who are all DU groups that we call weak DU groups can be diagnosed by the DU testing. Okay, so here I want to tell one important thing is called that if any person is say that I'm a negative group, don't say that you are negative group. You should confirm with the further testing. Should do a DU test. If the DU test shows that is a negative, we can surely say that you are a negative. Okay, until and unless you don't, if you are not doing the DU testing, don't say you are a negative. So this is why in the blood bank itself, what they are doing actually, if any report is comes as a negative, they are doing the another important test called what DU testing. Confirm with the DU testing and clearly tell about the patient, yes, you are truly negative. And one more important thing is about the people who are not have a chance of getting the D characteristics that they call D antigenic characteristics, simply called as the RH null syndrome people. RH null syndrome people say that actually they are not having the D genetic characteristics. So in absentee of D characteristics, simply called as a RH null syndrome people. You can see here actually this is about the RH blood group system which is surrounded in the RBC. So I told already RH is going to be D. Okay, so and here about the genetics mapping for RH system, which is going to be given a detail of three important scientists. One is called Fisher and Race, Wiener and Rosefield. And these people are giving idea about that RH system is going to be carry some genes. So the Fisher and Race give the idea about that we are RH system we are having. That RH system is composed of so many of genes. D gene, C gene, E gene. And these are the five genes are very, very important to contribute for our ABO grouping system as well as along with that your RH system. Without of all these groups, we cannot able to prepare your anti-D. If I wanted to prepare my anti-D, I supposed to need these CD antigens. So this is going to be called what? The genetic characteristics, how it is going to be expressed by the D group systems, help of the CDE genes. Okay. So in the examination for the practical process, example, we are trying to say that person is who's a positive. How you are telling, we are not taking up the all these five characteristics of antigenic. Only we are taking only D. So D antigen is going to be supposed to evaluate in the laboratory and saying the person is positive or negative. Let's we go for a pre-transfusional investigation. So investigation of any blood before a blood transfusion simply calls a pre-transfusional investigations. Okay, let's we go for what are the things that are there in the pre-transfer investigation. So ABO blood group RS systems. Before we transfer the blood to the patient, you must to check with all four-level investigation. One is the ABO grouping RS system is matched with each others. Number two is cross-matching methods match with each others. Coombs test or anti-immunoglobulin test is investigated as a negative for reporting. Antibody screening shows that there is a compatibility between the each others. If you do the four-level investigation, that give the enough idea about that, it's going to be a called safe transfusional testing. Let's now talk about the testing of blood groups. So if you are going to do the blood groups testing, we have the different methods here actually. One is going to be a blood group is going to be generally done by the what you call experimental methods called what slide method. This is going to see always for the maybe if outside camp the people are doing, they refer for the slide blood group systems. When you go for a hospital wise for a blood bank itself, they are doing the investigations called what spin tubing method. And this spin tubing method simply called what forward cell grouping method and reversal cell grouping method. And one more important thing is about that we go for a what some confirmatory information from the blood group system. They go for the conventional methods. So the conventional methods called what gel card method. So gel card method is an important method to come to know about that exactness of result of ABO group. And now they are modified into that conventional the automation method is going to be called micro well plate method. Is going to be very accuracy compare with the previous methods. And we go for the last method about that molecular genotyping method. What is that molecular genotyping method? Always we are doing the blood group with the slide and tubes and telling about that reaction. And here about that, I told already, we have a genotype and phenotype. Parent is having a A gene, B gene. I am producing my O groups. So this is going to be called as a what parentage gene we are having. So the parentage gene is supposed to be identify the molecular techniques, and this is going to be called molecular genotyping, which help you to come to know that PCR method. The PCR method will help you about that what antigen gene we have in the your blood cells. Let's talk about the first things about before we go for investigation of blood group system. We must know about that how the antibodies are going to be prepared in the laboratories. So they are having antibodies as a anti-seras. So the anti-seras means nothing but preparation of antibody from the antigens. So the source of antigen, how it is actually. We have a two different kind of picture here actually. You see the left side. This is going to be called what polyclonal antibody production, and another right side is going to be called monoclonal antibody production. We have a two different kind of anti-seras in the lab. You can see that actually polyclonal antibody, which is supposed to have the uh, A and that we call the AHG, anti-immunoglobulin testing. For investigation of anti-immunoglobulin testing, we are supposed to use that polyclonal antibody reagents. When you're supposed to go for ABO grouping, you can see that always right side monoclonal antibody we always use for the what A group, B group detections. Let's see about how the polyclonal antibody is going to be produced and given to you in the laboratory. You see that actually AB group of system we are using the monoclonal. For cross matching Coombs test we are using the AHG. That AHG reagent is prepared by the technical what polyclonal antibody production. So by the help of polyclonal antibody production, they enter the antigen into the rabbit. The rabbit 1 week after producing the antibody is supposed to be collected, and this serum collected things as a called as a purified protein complex, and this purified protein complex called what? Simply say that anti-immunoglobulin complex. Okay. The same way, when you go for a monoclonal antibody, the monoclonal antibody something is different. You can see that actually one single antibody is going to be separated out from the cell. It is going to be examined for the reporting. Now let's see about that. What is a mean of monoclonal? You see that from the mice, they are taking the stem cells like spinning stem cells, and they introduce a stem cell into the cancer part that is going to be a myelomal cancer preparation, and the cell is going to be myelomal cancerated after, and this is going to be called what myeloid cancer cells. And these cells going to be doing the cell culture in the laboratory, and finally, whatever antigen I wanted to introduce into that cell, automatically they are introduced. Example, if I introduce the A antigen into that myelomal cells, automatically the A antigen is carried into the cell. If I am introduce the B antigen into the cell, it is carrying that. And finally, we can see that whatever antigen I'm just enter into the myelomal cell, the myelomal cell will be separated out as A cell, B cell, C cell. I can prepare a separate antibody from this cell culturing. This is going to be called monoclonal stream culture. Okay, when you come into the first characteristics, it is produced by the rabbit. All the antibodies are mixed together. That is called polyclone. Multiple antibodies are present. In the case of right side, you'll see that the cell is separating the individual antigens. The individual antigen is supposed to be cell cultured. It is giving the antibody separate as A antibody, B antibody, or whatever something. So you'll see that actually when you see into your blood group reagents, A reagent, B reagent, they always mention that is called monoclonal antibody. That means they prepare by the myelomal cell culturing. Let's see the picture here. Actually, you can understand about the first thing about anti-sera A, okay, which is going to be simply called as a monoclonal anti-sera. So by cell culturing method, they are preparing anti-sera A1, anti-sera AB. You can see that actually anti-sera AB is going to be called monoclonal, but in the case of anti-sera A1, we always count this as a lectin antigen. So lectin anti-sera is nothing but it is performed by the that we call plant cells. Okay, and Dolicus bacillus seeds they are taking, and these seeds extract is going to be called as anti-sera of A1. When you come into the H antisera, the H antisera also called as here from the plant Urex europros plant. From the plant they are taking extracts. From the plant extracts, if you are taking any complex, this is going to be called what? Lectin antisera. You'll see that actually maximum of antiseras made by the monoclonal, and some of the antiseras which is produced by the lectin groups that we call plant extracts. The last one using as the anti-immunoglobulin, which is going to be called AHE reagent, which is are called as a polyclonal antisera, which is made by the, I told already, by the polyclonal method of antibody productions. Let's now talk about the anti-seras. If you are buying from a company, you must know about one important thing is about that anti-seras, whether it is quality good or not. We have to check by the quality, simply call what the important parameters, say that appearance, specificity, avidity, reactivity, potency. So what is mean of appearance? When you just open any ABO grouping kit, see that actually the kit itself have a proper turbidity. If it is absence of turbidity, absence of any particles. If it is there is no particles, if it is a clear appearance is there, mean you have a good appearance of age. Second thing is specificity. You have to know about that whether the reagent is specific to the particular antigen or not. You should check it. Example: A antisera. If you are taking your taking your blood group of A and checking with that whether it has to give a positive, we say that is a specific. And what is mean of avidity? So avidity means called what? When you are taking the any blood cell into the antisera, how long it take reacting? The reacting duration time is going to be noted in a second. This is going to be avidity time. Avidity normally will say that says 3 seconds, 5 seconds, 6 seconds, something. It has to maximum give the minimum second to get a reactions. Okay. And reactivity. Reactivity means whether it apart from the agglutination, which give you the Tolex formation, any other thing is going to be noted, and you should have to say that it's a good reagent or not. We compare with the reactivity tests. And finally, we go for potency. The potency is to come to know about that antigens which is present in the people, low quantity or high quantity, whatever it may be, it has to give some titer value. The titer value of antigen is given by this potency method. It help you about that. What is your anti-sera potency? Okay. So it's all going to be called as a quality control system. Before we run any practical, we always investigate all these 5 level tests in the sera and confirm that whether quality is good or not. Let's go with that chart. This chart is telling about that how the anti-sera is going to be tested in the laboratory. So if I take any anti-seral example, we'll go with the first one, anti-A. I'm taking anti-A in laboratory to testing that whether it's a good to use for a practice or not. So you say that actually we are taking the A1 cell. If I'm taking a A1 cell, anti-A is added to the A1 cell, it has to react. Definitely is reacting. The thing is actually you'll see that actually titer level is going to be about 250 level, and also the reaction is going to be appear within a 3 to 6 seconds. If it is happening within a 3 to 6 seconds, if the titer level is above 256 titer level, it shows that it is a good reagent. Your anti-A is going to be called what? Good quality. The same way, anti-A is tested with the A2 cell, A to B cell. Same also for anti-B, we are checking with the B cell, A to B cells. Anti-AB, we testing with the A1 cell, AB cell, A2 cell. So help of these all antigens is checking with the anti-sera, come to know that whether it is reacting or not. It's all going to be called what? Before investigating, you have truly check for anti-sera quality control. Okay. So if you are doing any investigation, definitely should be documented. Like same way, if you are doing any anti-sera quality control, you should be documented first. We'll see here actually. This is going to be called what's a worksheet. And this worksheet giving the idea about that whatever I done for a quality control, I am going to measure that all kind of documentation into the paper. This is going to be a worksheet paper. That paper will tell about that. Example, we see that the picture about anti-A, anti-sera. So you can see that the result of A anti-sera appearance, turbidity, precipitation, particle is nothing presence. Specificity reacted with the 3 to 4 plus amount. Reactivity is nil. Avidity by 3 to 4 seconds, and potency is 1 is to 156 for A1. So I can see that actually maximum the report shows that the excellency of A1 anti-sera with the antigens. So I can say that this the reagent is now in the quality. good quality. So we can go further for the investigation using this A1 anti-serum. Let's now talk about the such things about that blood group system is always done the laboratory. So as you know, all of you about blood group system by slide method we are doing actually. So the first thing is about that if you are performing a blood group system, you come to know that what should add first. Always we have to add anti-sera into the slide. Okay, just take these three slides and make the slide as a one is called as a and one called as a B, one called as a RH. And these three circle we made it, and these three circle we are adding the anti-seras. So what are the anti-seras we are having actually? A anti-sera is going to be a uh, blue color, B anti-sera is going to be yellow color, RH anti-sera is the colorless. Adding also all anti-sera into the one drops us into the each circles data. And finally, we taking the samples from the people finger blood or either a EDTA blood. We are adding into that. Finally, mixing this both complex. Wait for a 2 minutes. Within a 2 minutes, you should get a reporting of that reaction. That means called agglutination, no agglutination. Look at here actually if the person is report shows here actually A, B, RH. See that actually person is having A is agglutinated, B is agglutinated, and see that last RH system is not getting agglutinated. So say that actually the report of these blood group is AB negative. So that A antigen is present, B antigen is present, RH antigen is absent. So our anti-seras combined with the patient RBC, what the patient RBC having the antigen, it's supposed to be reacted, and this is going to be called as antigen antibody reactions. Okay, so this is going to be example of how the AB negative groups comes in here. Let's talk about the reporting of ABO anti-group systems in the slide method. Always you come to know that if you are giving a report to the ABO group system, you always give the report as a grading system. So here the grading system of 4, 3, 2, 1 plus or minus 4. So the meaning of 4 is going to be called what? High agglutination is happening. This is going to be that called largest strong agglutination techniques. If the thing is 0 is going to be no agglutinated, going to be 0. Okay, and these are plus or minus going to call it as a what? It's a antigen is supposed to be presence in a minor quantity. Simple called as a weak antigen. If person who's having a weak antigen, either it is maybe plus or minus report. Look at into the right side. Actually, see that example. You see the first group person is having the A positive. How you are telling the A positive? Because A is agglutinated and B is not agglutinated and D is agglutinated. That's why we call it as A antigen is present, T antigen is present. He is supposed to have a A positive. And see that actually the report itself we have a grading. A antigen, A antibody reacted in the 4 plus amount. B antigen, B antibody not reacted. That's why going to be 0, going to be called what? Grading is 0, and say that RH anti-systems are giving the 4 plus. That means it is reacted in a strongly. We can say that he is having a strong of A B anti, and it's going to be supposed to be called A plus. Let it go in for second case is going to be called as a B positive. So look into the second case. B positive is B is reacted and RH is reacted. That's why called as a B positive. Third people will say that actually AB people AB positive, A reacted, B reacted, also RH is reacted. That's why AB positive. Let's see in the last picture. The last picture shows that all negative is going to be called O negative. The meaning of O negative is no absence. There is absence of what? A antigen, B antigen. So absence of B antigen, along with that absence of H antigen, simply called as a O negative people. Okay, I told already if the person is O negative people, we should have to confirm with the further testing. It's called what? DU testing. So DU testing is very important. Come to know that they really a negative people or weak D positive people. Let's now go for another thing about the forward blood grouping, reversal blood grouping. Here the blood group system is done by the important techniques called what? Cell grouping and serum grouping. I told already we have a cell is having an antigen, plasma having an antibody. That is we are using here as a tool. For forward grouping, if we are doing and come to know that what's antigen we are in the cells. Reversal group you are doing with the plasma, you can say what antibodies we are in the plasma. So for investigation of these spin tube method, we need a patient RBC, we need a plasma. Without that, we cannot able to do it. You see here actually, before we go for performing the investigation of any blood groups in the tube method, we should do the what cell washing preparation. See that actually cell washing means nothing but we remove the unwanted protein complex from the cell surfaces. That's why we are doing the process called 2%, 5%, 10% cell washing. Here I'm giving some example to you how the cell washing is done. First of all, we calling the people and collecting the blood cell from the people that we call EDTA blood, whatever you take it. So you take a test tube and there put a one drop of blood into the test tube. So add a saline. Saline we have to add a 3/4 into the tube. Finally, the blood is centrifuged for 3000 RPM for 1 to 2 minutes. Finally, you are getting the what supernatant and the bottom it is having a deposit. So what we have to do, remove the supernatant that they call the top portion, and finally we go for a what repetition of these steps is adding the saline for a 3 to 4 times and wash that. So if you are continually washing the RBC for a help of saline, this is going to be called as a red cell washing. And this red cell washing is if you done, finally we are going to add a saline into the last point, and this is going to be a percentage thing. It's simply called as a what percentage suspension solution. Look at here actually about that. How will you form the 2% of suspension? If you are going to prepare a 2% of self suspension, at the end of fourth saline cell was, we are adding a one drop of blood. Already blood is there at the bottom, we have to add a 49 drops of saline. If you are adding a 49 drops of saline into the one drop of blood, finally, this is going to be a 2% self suspension. If I am going to prepare a 5%, one drop of blood along with that 19 drops of saline I'm adding, this is going to be a 5%. So end of our saline washing, see that bottom is our cell. The cell we are adding into the saline, whatever the amount is important, 49 drops we are adding, this is going to be a 2%. If I am adding a 9 drops, this is going to be 10%. Probably you can see that in the laboratory they are using the 2 to 5%, either 2%, 5% cell wash they are going to use for investigations. Okay. So next thing is about the microplate gel card method. They are always using the 1%. So 1% you are going to perform with the saline suspension is a higher quantity. Let's talk about the blood grouping forward method. So here about the cell group is if you're going to do in the laboratory, you must have this important thing is about prepare a 5 cell suspension from the who the donor are. Okay, if the donor want to know the blood group, you have to go for a forward blood grouping. Collect the blood from the donor EDTA blood, and you have to prepare, as I said in the earlier PPT, how to prepare the 5 cell suspension that is called tomato red suspension. Prepare the 5% cell suspension first of all, and keep ready for the red cell, also keep separate for the plasma people. Now you have investigation how to do that. So you have to take the test tubes made as a tube as a B, and D. And these three test tubes are called as A cell, B area, O D area. That is called, we are going to add a 5% of red cell suspend into the three tubes in simultaneously. We taking help of the pipette, we are transferring the one drop of A antigen, B antigen, that is called A cell, B cell, D cell into that region. After adding the resel suspend to the three different tubes, we are having the what the anti-sera. So the anti-sera what we are having? We are having a anti-sera, B anti-sera, D anti-sera. Adding this A anti-sera, B anti-sera, D anti-sera in respective tubes. Finally, mix the patient 5% red cell suspension with the anti-sera, and you have to leave it for a room temperature 5 to 10 minutes, getting a reaction. After 10 minutes, it has to centrifuge for a 1 minute. 1 minute after, look at to that actually see the bottom. If the bottom shows that any reaction, you have to say that whether it's a positive or not. Example, we see the button formation. If the button formation show that positive, look at that actually the pictures telling about A and B and D. A is getting the button, B is not getting the button, and D is getting the button. Yes, it is supposed to be A positive. Okay. So how to confirm the people again with the second method called what? Reversal method. The reversal method telling about that if the person is blood group is antibody, the presence of any antibody in the person blood, we can find through by the adding into the anti-seras, adding anti-sera into the plasma. Finally, we are telling about it. So this is going to be a reversal blood group systems. So we'll see about here. After getting a report of ABO forward reversal blood groups, we are taking as a reporting. So let's see about that. We are taking a ABO group system, which RS system, with the help of T. One is called forward grouping and reversal grouping. We'll see that actually this is going to be a called agglutination grading chart. The agglutination grading chart is help you about that. You are going to determine the correct report of ABO grouping by the help of interpreting report properly. I told already in the slide method, we are telling about 1 plus 2 plus 3 plus. The same way, you should have to interpret the report of ABO grouping in the test tube itself, presence of reaction. If the presence of button formation is going to be a 4 level, button formation lesser amount, it is going to be 3 plus. So according to the formation of agglutination, we are giving a report as a 4 plus to the 1 plus. Sometimes what will happen, I told already, it may be mixed report. Either it is a weak antigen is present, we'll say plus or minus, and maybe absence of any antigen, it is going to be, you can see that actually the last tube, you can see that there is no reactions. And look at to the example of picture here actually, how the people are giving as a O positive. If the O positive, how they are telling is action. You see that first tube, A is getting the no agglutination because he is not having a antigen. And B, second tube is not getting agglutinated. That's why called as a B is absent. See that actually third one is getting the agglutination. That's why you see the button formation. This is going to be a 4 level percentage is positive. So called as a RH positive. Okay, let's go for a serum group cells. If you go for a serum group cells, we are adding the anti-seras, that is called known antigens into the plasma. The plasma is having any antigen, it is supposed to be reactive. You see that actually the the fourth tube and fifth tube getting reacted. The fourth tube, fifth tube is getting reacted because of they are having anti-sera. So anti-sera A, anti-sera B reacted with the patient RBC. It gives as a what antibodies present. So finally we conclude that actually O positive. How we are concluding the O positive presence of antibody of A, B. We supposed to call it as a group of O. So this is going to be confirmatory for the people come to know that ABO grouping, forward grouping, reversal grouping. So forward grouping identification of antigen, reversal grouping identification of antibody. Here we identify that the person is not having antigen, the person is having an antibody. The antibody is supposed to have a antibody, B antibody. That's why we call this a O positive people. So this is information about that. How forward blood grouping giving a help to you, how the reversal blood groupings help to you. Actually. Let's go for another thing about testing of RS systems. Especially, we say that DU testing. So this is the DU testing call I told already. If the person any report shows that RH negative, we immediately go for a DU testing method. This is going to be a anti-sera we are adding into the test tubes. Okay, finally we are adding the person who says a negative. The 5% red cells have been added to the tube where we are going to incubate for 30 minutes and doing a centrifugation for 1 minute as 1000 RPM. Finally, you see that agglutination. If the agglutination does not appear, we are adding a 2 drops of anti-immunoglobulin. So anti-immunoglobulin added to that. Again, we go for a centrifugation 1 minute for 1000 RPM. Finally, we are examining agglutination. If the exam after that agglutination is present in the tube, it's truly called as a positive people. You can write the report DU positive. That means the person is having a weak D antigen. It is supposed to be reacted with the anti-immunoglobulin. So this is why called as a DU testing. You're supposed to do any people have a RH negative. You have to do this test for confirmation of weak antigen of D. Next, to go for a false positive reports. False positive possibility. I told already positive is going to be will get it. Sometimes what will happen, we are giving a false positive to the people because of your reagent is not good. If your contaminated reagent you are using, your suspension is not prepared properly. Maybe you are doing your high speed centrifugation. It are all giving you the false report of any blood group system wrong. Sometimes false negatives also will happen when you are using the maybe the error adding a anti-sera into the another tube. For example, A tube you are adding the B reagent to the A tube. This is going to be a collapsing of anti-sera into the different tubes. It make to the confusion of what your error report of negatives is. So you should avoid the ABO grouping positive negativity because of we have to do the proper protocol for the investigations. Let's next we go for a gel card method. So what is gel card method? So gel card method is simply say that we are performing for ABO group is precipitating. Okay, here we going to the precipitation of ABO grouping in a proper manner. So this is going to be called what? Trapping. So the trapping method simply called as antigen antibody reacted. As I said earlier, antigen antibody reacted is forming a button formation. It is giving the agglutination. And here you are not able to see any agglutination in the picture, but instead of that, we are seeing the trapping. You see that first picture, the positive means you can see that antigen antibodies are trapping in the top. If the report is a negative, you can see that is antigen antibody is not reacted, it has come to the bottom. So this is going to be called negative reporter. The first one called positive reports. So how to trap the antigen antibody? Because of presence of antigen antibody, they are getting a reaction as positive and negative. Okay. Let's go for a preparation of blood group system for a gel group method. You must know about that. One important thing is red cell suspension for blood group system for a card method, gel card method. We used to prepare 0.8 percentage. Okay, so you can see here actually. So 1 ml of a list solution, we are going to use it, mixing with the 10 microliter of uh, RBC and going to be prepared as a 1%. So here is going to be called what? Normally we are using the normal saline. Instead of normal saline, we are using the chemical called what? Low ionic strength solution. This is going to be should be used in the gel card method instead of normal saline. So when you go for performing the gel card method or whatever higher quantity diagnosis steps in the laboratory, don't use the saline. You must use the low ionic strength solution. It help you the what? Proper osmotic reaction. It give the proper reaction into the reporting. Okay, that's why they are using the what? LIS reagent. Let's now go for a testing of ABO grouping by the card. Okay, they are giving as a card to you. This card is going to be have a B, D, and one control and A1 and B. Okay, what we have to do actually? So we have to take the blood of patients. I told already 1% of red cell we are preparing, and also they are giving as a reagent. The reagent is simply called as a ID diacyl A1 B reagents. Okay. So first of all, we see that actually the ABO group system is going to be reported by the antigen antibody reaction. Here we see that actually ABD already is have the reagents. Okay. Only thing is we have to add a patient sampling. So patient sampling, you say that 10 microliter of patient red cell suspension, we have to add into the ABD. And finally, we are adding the patient plasma into the fourth one, fifth one. So waiting for a 10 minutes. After the 10 minutes, we are doing for a centrifugation. Here we can see that actually this is going to be a micro centrifuge incubation. Say that the first picture telling about that how to incubate this gel card by the help of special incubator. And the lower one picture is telling about that how to centrifuge this gel card in the special centrifuge called micro centrifuge. After we done the centrifugation with the micro centrifuge, see the reporting pattern here actually. Now we see that example of the person how is A positive. So how if the person is A positive? See that first picture, the first tube is getting the trapping, second is not getting a trapping, third is getting a trapping, and fourth is control is not getting a trapping. Okay, and finally fifth one is getting not trapping, and seventh one sixth one is getting the trapping. So say that actually how the person called as A positive. So A positive means he is having a antigen. That's why the first tube is getting the trapping, and positive means he is having the what RH positive. That's why you can see the third tube is getting the trapping. And how about the last tube? Why the last tube is getting trapping? Because of he is having the antibody. The antibody, if the person is having the A, he is having the antibody of B. So that's why the B antibody is supposed to be present. That's why you show that the sixth tube itself have a trapping of antigens. Okay, so this is going to be called what? Gel card method always say as a trapping techniques. Okay. Let's see here actually, you are giving interpretation for the gel card method always give as a reaction value. One is called plus value, 4 plus value, 3 plus value, 2 plus value. I told already about that trapping in the top of the tube called 4 plus. If present in the bottom, truly called as a negative. Okay. Sometimes what will happen, you are report getting some error due to the hemolysis, due to the mixed reporting. You should have to eradicate problem of hemolysis while we doing a centrifugation of sampling. You don't try to go for a high speed centrifugation for the process of cell washings. Okay, let's see about here actually. This is going to be a example here. A, B, one is a positive control A1B. and the groups of the person is B is getting 4 plus and A1 get 4 plus. Remaining all other things is are negative. Say that actually the person belong to the B negative. This is example of gel card report. How you supposed to be interpret. Okay, and this gel card also going to be used for the additional purposes. Example, same gel card going to be used for the anti-immunoglobulin isolation. We'll see that actually the first previous picture about that ABO grouping gel card, and this gel card is going to be a ABO blood grouping along with that AHG gel card. So this is going to be predominantly used for the anti-immunoglobulin investigation for Coombs test. So we can also use this Coombs test instead of manual method. They go for a what gel card method, helpful for the what proper finding our report. So you can see that this is going to be a newborn baby reporting. The newborn baby report shows that AHC is a negative. That's why the baby is not having a sensitized cell in the reporting. Let's go for another thing is microplate techniques. So microplate agglutination method. You can see that blood group is done by the microplates. You see that actually this is called YouTube microplates, and this microplate is going to be have the important thing is you call what agglutination. So the presence of agglutination in the tube itself, we can see that picture. The first picture is telling about that segregation of antigen antibody reaction in the entire tube bottom. If the reaction is not happened, you can see that accumulation in the bottom is going to be a negative report. So negative report, positive report, see the comparison. One is again not having a segregation of antigen. One is having the antigen antibody segregation on the tubes. So this way you're going to tell about the positive reaction, negative reaction. As I said already, ABO grouping, how we have done in a manual method, you have to do the same method here. Actually, instead of manual method, we are running with the microplate method. Micro amount we are going to take it, and finally the mission is evaluate the blood group. What is that? Okay, let's see about the next thing about how the reports will get by the plate. So this is going to be microplate agglutination reporting. I'll just inform you about one group reporting. See that actually there is a different 12 different patient reports here actually. So the first row going to be called A anti B, A anti B, A anti A B, A cell, B cell, D cell, and control. And finally, you'll see that actually the person is going to be a positive. How the first column is getting a positive means the person is having the antigen in the A as well as antibody in the B, and is getting a reaction in the absence of other thing is presence of D. So you can see that person is going to be a positive. So how the people are concluding by the gel card method is presence of that we call button formation, absence of button formation. According to the presence of button formation, it truly saying that what the report of belong to. Okay, we can see that the first picture is getting the what A agglutinated and also AB is agglutinated. That's why we call it as A positive people. Let's move on to the next picture about that. This is going to be called genotypic method, simply called as a next generation sequencing NGS technique. If you go for NGS technique, here we have a vision. This is called NC generator techniques. This is going to be identified the gene extraction. Gene is going to be extracted, gene going to be sequenced in the mission, and finally you are getting the grading of what genotypic characters. Your genotypic characters is going to be performed by the PCR method, by the method called what gene sequencing methods, simply called as a generation techniques, is going to be advanced mission in the blood banks. Okay, so by the help of these method, they are going to performing the genotypic characteristics of blood grouping. Okay. Let's go for a cross matching. So cross matching is called compatibility testing. The compatibility testing simply called as a compatibility between whom? Compatibility between the patient and between the donor. It's going to be called what compatibility testing. Here we're going to do the compatibility testing by the major cross matching, minor cross matching. What we have to do for major cross matching? Donor red cell, patient serum, we are adding together, and this is going to be a minor cross matching. Uh, Major cross matching. Minor cross matching is a donor serum and patient red cell going to be added. This is going to be a minor cross matching. In both category, you will see that actually mixing of one red cell, mixing of one plasma, we'll come to know that what is antigen antibody reacted. Say that they have a connectivity, they have a agglutination, or they have a compatibility or incompatibility. Okay, here we're going to use the blood samples, EDTA sample for the investigation of cross matching, ABO grouping. We see the example for the major cross matching by the single tube method. If you are going to do the single tube method cross matching, we're going to do the phase techniques. One is we call what, using the saline, we're going to run it, and we're going to run with the albumin, we run with the anti-immunoglobulin. So by help of these three phase, if you are doing the investigation, is going to be called what, single tube method conventional techniques. Okay, how to run this method of cross matching? See the first procedure, saline cross matching. When you are supposed to do the saline cross matching, you have to take this patient serum samples, okay, 2 drops, and we have to prepare the donor red cell 2 to 5% red cell suspension and put the 1 drop into the test tubes. We mix together that we call 2 drops of plasma of patient donor red cell mixed with the test tubes. It is incubated for the room temperature 10 minutes. After it is centrifuged for 1 minute, we'll see that actually if there is no agglutination, it's going to be compatible. If it is agglutinated, if it is going to be a chance of presence of IgM in the antibody. If it is agglutinated present, we can say report is got incompatible. So that if the report is negative, don't say that is going to be compatible. You go for the further call, as you call albumin phase. Whenever you say that the report positive, you can go ahead with the agglutination or incompatible. Okay, if the report shows that no agglutination, you go for a further testing called what, albumin phase. How to go for albumin phase to confirm it? Let's go for a thing is about albumin phase. So if we are going to do the albumin phase, the first thing is about that we are adding the tube is already we done with the negative cases. The negative case report has been continued for the further testing. Add a bovine albumin 22% into the sampling, whatever we done previously negative. The negative report is have been continued with the phase 2. Adding the 2 drops of 22% bovine albumin into that, and finally we are incubating that. After incubating, centrifugation for 1000 rpm for 1 minute, and see that hemolysis. If the hemolysis presence, you say that it is going to be agglutinated, it is incompatible. If it is not agglutinated, you don't perform the report as a negative or report as a uh, uh, compatible. You go for a third step is called what? Phase 3 process. Phase 3 process means the negative case report has been done by the third level of investigation. So what you have to do means after report gets negative, the cell again to be washed with the saline 3 to 4 times, and we continue with the what next phases. Okay. So the next phase is called what? Anti-immunoglobulin method. So the anti-immunoglobulin method simply called as a AAG method. Okay. If you are performing the AAG method, the third step is adding into the 2 drops of anti-immunoglobulin and centrifuge the samples for 1000 rpm for 1 minute. And finally, you'll see that actually in the bottom, if the bottom shows that agglutinated, say that is truly incompatible. If the report shows as negative, it is compatible. So the steps 3. First step is we done with the saline method, we done with the anti-immunoglobulin method. Third step is done with the AAG method. All these 3 methods shows that as a negative, it's purely called compatible. If it is agglutinated, you say that is incompatible. So this is the clarity to confirm about that. Before giving a transfusion of blood, always check that whether all these 3 steps be done or not. One, saline phase, albumin phase, and anti-immunoglobulin phase. Okay, now let's go for a cross matching for emergency period. They'd always do for a emergency condition, and in accidental cases, they try to do the fastest method of investigation. See that actually 15 minutes process. So 15 minutes process, they're taking a patient serum sample and 2% of list patient RBC they are taking, and finally they incubating for 15 minutes for 37 degrees Celsius. They're doing a centrifugation for 2 minutes and see for hemolysis. If there is no hemolysis, adding into the anti-immunoglobulin finally look for the agglutination. If the agglutination is absent, truly called as a compatible. If the agglutination is present, is incompatible. Okay. Presence of agglutination is incompatible. Presence of no agglutination is compatible. Finally, say that the people blood is matched. So this is going to be called what, cross matching done in the laboratory in emergency conditions. Okay. Let's see about the picture here. Actually, how the major minor cross matching is compatible because there is no reaction. The end of 3 level investigation, you are saying that is a negative cases. See that another picture is tell about that incompatible. See that both tube is getting the bottom reacted, so the blood is not compatible. Don't give the blood to the patient. Okay, so this is going to be called what? A compatibility incompatibility testing by the blood group RH typing method by the cross matching techniques. Okay, see the picture here in the about the different types. Okay, how the blood groups are compatible with each other. If say that if person is A antigens, example, you say the donor O groups, donor O group is combined with the other groups, we'll always have some compatibility. I told already, ABO grouping is going to be matched with the what the known groups is. Always O is going to be a initial donor, AB is initial recipient. Though you have initial donor, initial recipient, you always supposed to do with the what compatibility testing to confirm whether it's a compatible or not. Okay. Let's now go for a procedure for the issuing of blood. Before giving a blood to the any patients, first of all, we must to say that whether it's a correct register number, correct blood group system, whether collected properly, the date of collection is correctly, there is no expert and you see that actually there is none of positive for testing of infected diseases, whether the sample is properly crash matched, patient blood group is properly checked. You see that all the thing is very necessity for before giving a blood donation, blood sampling. This is going to be important criteria to avoiding the blood transfusional wrong reactions. Let's go for anti-immunoglobulin Coombs method. Anti-immunoglobulin Coombs method, come to know about that. The person is having the auto antibody, allo antibody. What does it mean of auto antibody? Person is who's having the self antibody called auto antibody. Person is who's try to produce the antibody after induction of any antigens called what? Allo antibody. These antibodies is supposed to present. It has been identified by the techniques called what? Anti-immunoglobulin technique. So anti-immunoglobulin technique is going to be two methods. One is called what? TAT method, IAT method. What does it mean of TAT? TAT is called what? Direct anti-immunoglobulin method. That is to find out what the sensitized red cell in the body itself we have. It is performed by the method called what? TAT technique. And see that actually person who's having any antibody in the blood systems can be ruled out by the method called what? Indirect method. The indirect method is we try to do the sensitization of cell by the in vitro technique. So in vitro technique, we are doing the process called what? Indirect and immunoglobulin. Both method shows that negative. Truly say that the blood is compatible. Whenever before you go for a blood transfusion, always you sure about that anti-immunoglobulin method says that negative. It is going to be a safe donation. Okay. So here they're using the Coombs reagents. Always the Coombs reagents going with the anti-immunoglobulin IgG molecules along with that complementary system anti-C3D complex. So this is going to be important anti-sera we supposed to use for the anti-immunoglobulin testing. And here about the sample is EDTA we are using. Always you come to use that actually whenever you get the report of positive negative, you always cross check with the control cells. Here we have a positive control sample, negative control sample to perform the report whether your reagent is better on giving the reporting or not. See that the first thing is about direct anti-immunoglobulin method. The direct anti-globulin method, the principle is, I told already, the antigen antibody get reacted and finally it is exposing the reaction normally. And here is what the problem is actually. Antigen antibodies bind together. You are not to get the reaction, but is reaction is enhanced by the additional antibodies called what? Anti-immunoglobulin. See that actually autoimmune disorder people, that we call IAHA people, hemolytic disease of newborn baby, all these transfusional reaction cases, drug-induced cases, you'll see that they have the antibody in the RBC, but it's not reacted. This is going to be called sensitized RBC. And this sensitized RBC can be performed by the technique called direct immunoglobulin. Come to know that either patient or donor having the what? Autoreactive antibodies. So this is going to be ruled out by this method and say that they are having the some sensitized cells in the blood or not. Okay. Let's go for the investigation of that method if we are doing actually we are going to prepare a 5% red cell suspension. Along with that we are adding the 2 drops of red cell suspension with the anti-immunoglobulin reagent, mix that and centrifuge for 1 minute, and finally you see the examination of agglutination. If the agglutination is present, directly write the report as a direct positive. If the report shows that negative, go for a centrifugation and finally add with the positive controls whether it will confirm again whether it is reacted or not. If it reacted means your reagent is good, you can write the report as negative. Okay. So the positive report, negative report confirm properly after they're done with the control testing and say that truly positive, true negative. Okay. So next method called indirect immunoglobulin. Indirect immunoglobulin is going to be called presence of halo antibody, going to be identified in the laboratory by in vitro testings. Okay. So in vitro testing is going to be called as ICT. ICT is called anti-immunoglobulin indirect method. So here we are having a test tube with the 2 drops of patient serum and adding the 5% red cell suspension of donor. Finally incubate for 30 minutes, 37 degrees Celsius, and doing the 1 minute for the 1000 RPM rotation. Finally, we see the agglutination. If the agglutination is present, directly you can write the report as a positive. If it is a negative, you can go with the adding of anti-immunoglobulin, 1 drops. Again, go for a centrifugation, and finally you will see that the examination of agglutination. If the agglutination is present, we can write the report as a positive. If the agglutination is absent, we can write the report as a negative. So this is going to be give report of ICT positive. That means he is having a sensitized cells in the blood and he is having a particular antibody in the plasma. And he say that actually the source of error, how the TAT method giving the error, sometimes TAT ICT giving some false positive because of contaminated reagent, maybe over centrifugation, dirty glass items, maybe give the wrong reporting. Sometimes false reportings may happen due to the serum is not matching with that. Maybe the reagent is not properly you have taken. This is going to be a cell suspension not properly prepared. Maybe less cell centrifugation is going to be also make the false in the reporting of ICT and TAT. Next method is going to be called as detection of anti-seras. Example, this is going to be antibody is going to be detected in the large manner. It's going to be called as a what identification of blood grouping anti-seras in the blood system by the help of immediate spin method, albumin enzyme method, and direct immunoglobulin method. Help of these 3 techniques come to find out about the people who what are the antigenic system they're having, what are the antibody system they're having. So you can see here actually there are the 6 different antigens we are carrying in the surface of RBC apart from that ABO system. So one is called KEL, Lewis, Duffy, KIT, MNSP. So what is going to be happen actually? Person who's having the antigen, sometimes what happen, they are not having the antibody, but you can see that between the patient and donor, some of the antigen is present, some of the antibody present in the some people that may reacting with the people of what other blood group system may be interrupting the blood transfusion reaction. So you should have to perform the what antibody screening of what other than the ABO that in the laboratory is called what antibody screening panel test. These antibody screening panel tests give the idea about that rather than ABO group, what are the other antibodies we are having? Example, Lewis antibody, KEL antibody, Duffy antibody is there means if who person is having the KEL antigen, it is reacting with the KEL antibody, he is supposed to get the what hemolytic reactions. So always you come to have idea about that ABO grouping system not only getting the hemolysis, also other than the ABO group system also giving the hemolysis. So the hemolysis is going to be avoided by the performing the test called what antibody screening panel test. See here actually this is going to be antibody screening panel test. They are taking the what 11 different antigenic characteristics they are taking as a phenotypic characteristics and tested with the patient. You will see that actually one result here actually. The result shows that about person is having the uh, that is called is having the important antigens of RH, importance of rhesus groups and other things. So say that actually the person, whatever the antigen is present here, will give the positive reports. And according to that, we will say that how many antigens are there, how many antibodies are reacted. We surely tell about that matching with the each donor with the each patient. Example, see that this is going to be at 11 different patients. The donor sample is here actually. We confirm with the patient. Finally, we'll get the idea about that which donor sample is matched together. If the if the donor blood sample matching with the patient sampling, we'll say that what are the antigen antibodies are going to be connected. According to that, they say that actually it is matched sampling and unmatched sampling. So this is going to be called what screening panel for antibody. What antigen antibody they are having. If they are having a similar antigen and antibody, they do not give the blood. Okay. So these important things about that blood group system is very important. Come to know about that. What antigen antibody supposed to they have in the blood systems. Hello.
Yes, sir.
Sir, can I connect with the students now?
Yeah, yeah, fine, sir. I'll just ask them. Sir, there are no doubts. You can please proceed accordingly. Thank you.
Okay. Yes. So this is the report for now. I have some recalling things. Yes. Okay. Yes. Let's see the first things about the slide is going to be have a grouping report. So one is getting the anti A, anti B, anti D. The first picture is telling about that three important positive reports. Okay. And second thing is three important negative reports. So probably the blood groups belong to the first one is AB positive, and second one is going to be AB negative. So this is going to be example here I'm giving you. Yes. So let's go for the next interpretation. The next interpretation about that. See here is a two picture about that. One picture is going to be have one report. Second picture one report. So one first report is going to be a 4+ report. Second picture getting the negative report. Say that 4+ agglutination in the first picture. Second picture show that is a negative report. Now let's go for the blood group report, forward blood grouping. You can see that the tube is having the 5 test tubes. First tube, second tube, there is no reaction, so there's going to be no possibility of AB. And see that the last, the third one is going to be have agglutination, going to be positive. And fourth one, fifth one, A, B is getting reacted. B is having a anti-sera, B anti-sera. That's we called as a O positive. So the person belong to the O positive group here actually. Now let's go for a gel card method. So gel card method says that person is having the reaction in the B, in the that we call trapping in the B area, and also have the A1 getting a trap area. Say that is having the B antigen, A antibody. So say that person is having the B negative report. Say that how the B negative is happening. Say that RS system is getting the no trapping. That's why control report is negative. Also, test of patient is T report is also getting the bottom. So that's why the report says that B negative report.